Okay, so this is what we left off on last time. And so right here we have a engineered artificial photosynthesis using alternative CO2 fixation pathways. Now, I'm going to be honest with you guys. Well, you. This was a big field of research a bit ago. And it's a cool field of research because there's things that could be done maybe in the long term, like increasing the efficiency of photosynthesis in plants, or utilizing these to make cheaper or self-growing, self-repairing photosynthetic cells. But in terms of efficiency of the light that's captured from the sun, we've surpassed plants almost a decade now. Yeah. So now, for the light that's captured in the wavelengths that's captured, the plants are still very efficient, okay, compared to most solar panels. But we capture a lot bigger range. And so this is something that biochemists and biology people tend to be like, they don't understand that. And they always like to calculate the efficiency of the plants and go, oh, they're so efficient, and solar panels are so bad comparison. But we're capturing 10 times the range of them in a solar panel versus a plant. So, even if we're less efficient in the blue and green and red and yellow being the big ones, we're a hell of a lot more efficient overall. Yeah. But there's a lot of tricks that plants use that can be learned from. So it's cool, but it's not they always make it sound like it's going to change the world. And maybe like 100 years from now, yeah, sure, but yeah. No, it's not like a breakthrough and then fusion. It's not like that. So synthetic biology, using engineering principles to develop potentially useful devices. So what we're taking here is spinach leaves, which can be fairly easily grown. And then we're taking those, we're purifying the thioalkaloids. We're then joining them into structures that can then be used as essentially like solar panels. Though the solar panel arrays are not necessarily the most efficient way to do that. They're only that way because of the engineering consequences of the current. Yeah. Anyways, what it does is it actually, with the presence of NADP / NADPH, ADP, and the Setch cycle here, it actually is generating from CO2 glycolate. Now, that doesn't sound super impressive, but what that really tells us is that you've heard a lot about CO2 capture probably. It's been a big thing on the news. Have you ever heard of feedstocks? No? Yeah, it never gets talked about, but if tomorrow we took care of all energy, fusion snaps, and we can build them in a year and they'll be safe. Okay, boom, energy's taken care of. Okay, car energy taken care of. Bam, magic. We would still need to pump oil. Like 30% of oil, 30 to 40% depending on the year, is actually used as feedstocks to synthesize everything that you have around you all the time. Your desks, your chairs, your clothes, everything. Okay? Glasses, all of it. And so they're the base components we make everything else out of. They're those like assemble this from 3 and 4 membered rings and stuff that we're doing in organic chemistry. Those are the 3 and 4 membered rings. They come from oil. Okay? Almost exclusively now because there's just not any cheap places to get them. And so this is the sort of stuff that will replace that eventually. Okay? It's still in its infancy. It's still in the prototype phase. It's not anywhere near being scaled up yet. But it is getting pretty fucking close to being able to scale up. And I would say within the next 40 to 50 years, I would not be surprised if there is a 10%, 20% of that feedstock is coming from this stuff. So, which is actually pretty damn impressive and really fantastic, because this will actually lower CO2. By the way, I actually wonder if in like 100 years, we're going to be back to, okay, so did you know that in the 1970s, they actually thought we were bringing on an ice age? Yeah. Humans are bringing on an ice age. We're going to enter an ice age. It's all your faults. We should kill off a third of humanity, stop any energy production, and go back to the Dark Ages. Yeah. And so the exact same narrative was happening in the 1970s as currently is happening. Exact same stuff was happening, same conversations. The only difference was it was an ice age versus global warming. And that's actually because, and this was only recently found out in around 2010-ish, though it's only starting to work its way into the actual conversation, because there is the global warming conversation. Are we probably causing some global warming? Probably, honestly. But it has become so political that it's drowning out all of the actual research going on. Yeah. And in fact, it has gotten so bad where articles don't even get published anymore. They aren't allowed to even go to a review. Yeah. So it's gotten crazy. But I do wonder with technologies like this and all the capture stuff where it's synthesizing useful stuff, if in 100, 200 years, we're going to be talking about, we need to put more CO2 out. We probably will. Anyways, we should talk about it sometimes. It's actually fascinating conversation. Yeah. It's pretty cool stuff too. But anyways, these are the illuminated droplets here, and they'll be producing glycolates. Glycolates can then be assembled into a variety of different products, and then you could make, your makeup, your clothing, your glasses, whatever, okay, from glycolates. I already have stuff like this. Have you ever seen my plastic wheat straw stuff? It's the bowls and plastic-looking things I use. They kind of feel and sound woody. Yeah, those are wheat straw. Yeah. So this is already like a first sort of attempts on replacing plastics with other stuff that can decompose in the environment and won't cause, microplastics have been found in everything all over the world now. Yeah, they're in places where no human has ever been. Yeah, they're even below the Earth's crust. Interesting thing. Do you know that most of the water is actually way below the Earth's crust now? Yeah, they think that instead of meteors releasing water onto the Earth, it actually got brought up. Yeah. Fundamental rewrite of all geosciences. Yeah. Yeah. And they found water down in the magma where diamonds are formed now. Diamonds are capturing water. And so they finally found some diamonds with water in them. Yeah. Geoscience is having a moment. Really cool. Anyways, this is the artificial photosynthetic system. You make these little, essentially glass blocks, and the light energy causes this reaction to go. Theoretically, you could use this to make biofuels and put that in cars and stuff, but it's probably going to be more of a feedstock thing. We'll see though. The last time I saw any math done on biofuels, which was switchgrass, the math was that you would have to take up 80% of all arable places you can plant crops, just to feed the current car stock back in 2010. So you'd have to kill off 80% of humanity because we would not have food for them. Which is actually the current proposal, by the way. Do you know that? Yeah. So if you actually pay attention to the WEF and the major environmental groups, the current proposal actually calls for population numbers where 80% of the projected population growth would have to be killed to do what they want to do. So, anyways, just saying, maybe not the best thought out things, but I do agree something does need to be done. Make more innovation actually is the answer, but we'll see. I rarely get agreed with. The activity of the Kelvin-Benson cycle actually depends on environmental conditions. So the principal means of the regulation of fixation of CO2 into biomolecules is actually altered by the stromal environment by the light reactions. So light reactions actually lead to increases in stromal pH and stromal concentration, magnesium 2+, NADPH, and reduced ferredoxin. So all activate the enzymes of the Kelvin-Benson cycle in the stroma. So this right here, light regulates the Kelvin-Benson cycle. So here we're seeing this light regulation of the stroma Kelvin-Benson cycle. And so here it's dark. We have the thylakoid, magnesium 2+, H+, and we have magnesium 2+, H+, the NADP+, which is again an oxidized version, and here we have the F, which is oxidized. Now, in the stroma. Under light conditions, what's going to happen is we will generate NADPH from the NAD+, and this will give us the reduced form of that ferredoxin. That's what we've been talking about. This is just a much nicer looking graphic. In the stroma, the pH is actually going to increase from 7 to 8. The magnesium 2+ levels will rise. This results from the light-driven pumping of protons into the thylakoid membrane. Again, here, notice the numbers here. These magnesium 2+ big, small, decreasing. Increasing externally. H+, decreasing out here, increasing internally. And so the Rubisco activity is going to increase markedly on illumination because the carbamate formation is actually favored by the alkaline pH. The magnesium 2+ will bind to the carbamate to generate the active form of the enzyme. So the Rubisco activity is not directly actually affected by the light. That's not why it increases activity in light. It increases activity in the light because of this other pathway right here that favors the conditions which increase the activity. Okay? And that makes sense. Yeah. So reduced ferredoxin and NADPH signal that conditions are right for biosynthesis. Thioredoxin, it activates the biosynthetic enzymes, including chloroplast ATP synthase, when it's in its reduced form. The neighboring cis residue cycle between a reduced sulfhydryl and an oxidized disulfide form. And remember, the sulfhydryl, hydrogen on it reduced. Disulfide, two sulfides together, that means it's the oxidized form. So this acts by reducing the disulfide bridges that control the enzyme activity. It also inhibits several degradation enzymes by the same means. So this right here is your disulfide bond, that would be your oxidized form. When you put hydrogens across there and break that disulfide, that would be the reduced form. So these are the enzymes that are regulated by the thioredoxin. This is the list right here of the enzymes. This is the pathways that they'd be involved in. And you can see this is actually quite extensive. So in chloroplast, the ferredoxin, thioredoxin reductase, this dash is in between, by the way, and it got broken. This reduces the oxidized thioredoxin. This can be then reduced by the ferredoxin. And this is that cycle right here of oxidation or reduction. This is oxidized form. Light is then going to help intermediate that to the reduced form. And that's actually what we've been seeing up here. Okay? That's that cycle. All right. And so this is actually joined by via this ferredoxin thioredoxin reductase. And so this is the thioredoxin here. And so that's that thiol bridge, the disulfide bridge here, being reduced. And then this reduced form, this is then going to be oxidized here. And so notice that this is reducing this, allows this form then to react over here. And this is going to be the inactive enzyme, which has the disulfide bridge. We reduce that. That's going to give us the active enzyme, which is the reduced sulfurs. Okay, and then this will spontaneously oxidize via oxygen. So oxygen here is going to react with the reduced form, and that's going to oxidize the inactive to the inactive enzyme. All right, so let's talk about regulation via the NADPH. So the CP12, this is going to be an intrinsically disordered protein that's going to inhibit the enzymes through association. NADPH activates two biosynthetic enzymes by disrupting their association with the CP12. The phosphoribalokinase, glyceraldehyde 3-phosphate dehydrogenase. Those are your two enzymes. The C4 pathway of tropical plants and grasses is actually going to accelerate the photosynthesis by concentrating carbon dioxide. So because the oxygenase activity of the rubisco increases more rapidly with temperature than does the carboxylase activity, hot climate plants actually have to concentrate the CO2 at the site of the Kelvin-Benson cycle. The C4 pathway, the Hatch-Slack pathway, the metabolic pathway that concentrates the CO2 in bundle sheath cells, the major sites of photosynthesis. C4 plants, including tropical plants, grasses, and sedges. Interesting little factoid, part of the greening of the planet because of global warming / increasing CO2 actually has meant that these plants have to do a little bit less work. And so they're actually becoming a bit more dominant than they used to be. Yeah. And they're increasing their zones that they populate a bit. Okay. So the C4 pathway, this is the carbon 4, as you could probably guess. C4. Carbon 4. Yes. Chemists. Yes, we're so imaginative in our names. These compounds carry the CO2 from the mesophile cells to the bundle sheath cells. How much have you guys covered plant biology? Is any of this making any sense? Yeah, kinda. Good. Yeah, the important part is to understand the overall pathway. And this is one of those things in my evaluations next week. When we do that, you might want to let me know. Do you want me to cut more of this out, try to include other things? That's sort of things that I actually do act on those. For example, you guys may have noticed that I split the exams up. So decarboxylation of the C4 compounds maintains a high concentration of CO2 at the site of the Kelvin-Benson cycle. The 3-carbon product returns to the mesophile cell for another round of carboxylation. So this is that C4 pathway that concentrating the CO2 in a nice, beautiful little figure. So again, here we have the air. We're taking in CO2 from the air, obviously. This is going to be the mesophile cell here, and this is the bundle sheath cell here. So what's going to happen is this is going to be joined, the CO2, with the phosphoenolpyruvate to oxaloacetate. Anybody recognize that? That sounds a lot like something that would happen in the citric acid cycle. Yeah, so again, remember that these mechanisms are not like reinvented all the time. And these are very, very ancient cycles. Anybody know what came first, animals or plants? Common misconception. Almost everybody always thinks that plants came first. No, animals came first, as far as I've heard, though this is pretty new science, like hundreds of millions of years before. Yeah. Oh, yeah. So, yeah. Plants are actually the more advanced ones. And they have far more DNA than us. Corn can have 800, 1,200 chromosomes. Yeah, and all of our effort goes towards maintaining and growing corn. We even have special subsidies to maintain and grow corn. So who's the real ruler of this planet? Corn, damn it. All right. So oxaloacetate, this is then going to go to malate. Malate will then go to malate not in the mesophile cell, but in the bundle sheath cell. So we got transfer there. That malate is then going to release CO2 by going back to pyruvate. This is actually the CO2 that's going to enter that Kelvin-Benson cycle where we're concentrating CO2 so we can help to drive that cycle forward. Okay? The pyruvate is then going to get transported back to the mesophile cell. This is actually going to use an ATP going to an AMP. Note that this use of the ATP going to AMP is going to give you the 2-free orthophosphate, right? The PPI. This is going to then give you the phosphoenol pyruvate. Notice again, we transferred a phospho on there, didn't we? Okay. By the way, to transfer the phosphate, you end up getting PPI coming off of here. That means you have to actually incorporate orthophosphate. It's kind of an interesting, weird pathway. So anyways, this gives you the phosphoenol pyruvate that's going to get reused. And so you complete that cycle over and over and over. Notice, by the way, almost everything's a cycle in biological organisms. Anybody know why that would be? By the way, if you're listening to the video, this is the moment you pause, think about it, and try to build up some critical thinking skills. Why would you do cycles? Exactly. Yeah. So if you do a cycle, then you don't end up throwing it away each time, right? Kind of like, why do you build a meat hook to last? Or why do you build a shovel to last? Well, you don't want to have to build a new one every time, right? So the net reaction of your C4 pathway is this right here. CO2 in the mesophile cell plus ATP plus 2 H2Os. This gives you a CO2 in the bundle sheath cell, AMP, 2 free phosphates, orthophosphates, and 2 H pluses. The C4 pathway actually requires an extra 12 ATP when compared to the C3 pathway. And so it is actually quite energy demanding. Your net reaction of the C4 pathway in the Kelvin-Benson cycle is this up here. And so we have 6 CO2s, 30 ATPs, plus 12 NADPHs, plus 24 waters. This gives us sugar, one sugar. And 30 ADPs, 30 orthophosphates, 12 NADP pluses, and 18 H pluses. Now those 30 ATPs that are consumed per hexose in your C4 pathway, the 18 ATPs were consumed per hexose in the C3 pathway on the other hand. So this is much, much less efficient. Stomata, the openings in the leaves that allow gas exchange. When rubisco is adequately supplied with CO2, stomata do not have to open completely. This will prevent water loss, which is actually a major concern for most plants, right? Because most plants do not actually have a water table that would reach their roots, okay? And so they actually do have to store water loss, store water quite consistently. Now, an interesting little factoid, remember the greening of the planet and all that? Okay. Part of that's happening because plants in the northern hemisphere and where it's getting hotter have become less efficient because of the heat. And so there's actually been some struggles with plants doing this, and certain plants are not making the cut. So we're actually probably on the verge of an evolutionary change with plants. Yeah, which is quite fascinating. Though, for some reason, people think the evolution has stopped and there should never be more any evolution. So apparently that's a thing. But whatever. So this is both C3 and C4 plants are going to be important. One is more efficient under certain conditions. The other can survive other conditions. Does that make sense? So for example, if you planted these trees here in very arid conditions, they'll die. And they do die quite easily from arid conditions, don't they? Yeah. Whereas corn, on the other hand, if you put it in a place where it doesn't get any water for a month, it's actually okay. It actually needs very little water, right? Yeah, it can be very, very efficient with water when it wants. But it sacrifices that. it sacrifices that to the fact that it has to use less efficient pathways and uses more energy, more oxygen to generate that. Okay? And so that's what we're looking at. Many plants, including succulents, actually keep their stomata closed in the heat of the day. This is actually to prevent water loss. CO2 cannot be absorbed during the day. Instead, CO2 will enter the leaf when the stomata is open at night. The crassulous acid metabolism, CAM, this is the adaptation that the plants use to store CO2 until it can be used during the day. So basically, they open during the night, they do their breathing, store the CO2 then, while they have less evaporation to the heat of the day, right? And then they close, so they evaporate less, and then they're going to use up that stored CO2 during the day where there's light, okay? So at night, the CO2 is going to be fixed in the C4 pathway into malate and stored in vacuoles. During the day, the malate will be decarboxylated and the CO2 becomes available in the Kelvin-Benson cycle. And so you may have been wondering why the hell use C4? That's the C4 pathway. That's why the C4 pathway exists. That's why it's actually beneficial. Okay? Yes, the C3 pathway is more efficient, but you actually have to use it then and there. And that means you're probably losing a hell of a lot of water. If you're something like a cactus in the middle of the desert, you're dead, right? You can't do that. Because malate is the sole source of CO2 and malate storage is limited, the growth rate of CAM plants is slower than that of C3 and C4 plants. So pentose phosphate pathway, this is actually probably more important for you all because most of you just are not going to be doing a lot with plants. Though, to be honest, I do really think the plant stuff is incredibly valuable because those that don't go into the medical field, that's the money maker for biology stuff. Oh yeah, plants and insects. By far and away. If you want the best paid and best recruited job for biologists, do you know what it is? Specialists in insects. By far and away. Well, think about it. Like, what's the base of every ecosystem? Plants and insects, right? What pollinates most plants? Insects. What do most plants need somewhere in their cycle? Insects. What kills off most plants? Insects. So, like, of the biologists, the insect specialist is just, if the biologist gets paid $40,000, the insect specialist is at $100,000. Yeah. Far better paid. Far better recruited. Way more jobs. Pentose phosphate pathway. Again, the pathway is going to be generating a crucial source of NADPH for use in oxidative biosynthesis and for protection against oxidative stress. This is going to occur in the cytoplasm for all organisms. This is one of those ubiquitous pathways that tends to get conserved heavily. The first phase is going to be the oxidative generation of NADPH. This is going to occur when the glucose 6-phosphate is oxidized to ribulose 5-phosphate. What does pentose mean again? Yeah, 5-carbon sugar. And phosphate? You're attaching a phosphate, aren't you? Yeah. And so, look at that. We've gotten our name, haven't we? Woo. All right. And so, our glucose 6-phosphate plus 2 NADP pluses plus H2O, the ribulose 5-phosphate gives you ribulose 5-phosphate plus 2 NADPHs, which again is the reduced energy rich form, plus 2 H pluses, and finally CO2 here. The second phase in the non-oxidative interconversion of 3, 4, 5, 6, and 7 carbon sugars. Interconversion will rely on the same reactions that lead to the regeneration of the ribulose 1,5 biphosphate in the Kelvin-Benson cycle, which is why the Kelvin-Benson cycle gets taught before the pentose phosphate pathway, because they overlap in these, okay? And then this is why this is so important, because this is one of those key phases that you have to go through to convert your various different sugars into the different forms you need, right? Remember that a lot of these sugars are going to be incorporated and used for other structures as well. It's not just sugar to give you energy. The pathways that require the NADPH that we're going to be making are going to be these ones right here. These are the reductive biosyntheses. Here we have the fatty acid biosynthesis, cholesterol biosynthesis, neurotransmitter biosynthesis, nucleotide biosynthesis. Protection from oxidative stress is going to have the reduction of the oxidized glutathione, and the cytochrome P450 monooxygenases are going to use this. What is oxidative stress, by the way? Yeah, you have too many oxidizing species. And so this can then start oxidizing everything you don't want to be oxidized, right? Which, remember, oxidizing things is how you make energy. And it tends to be very energy efficient to do that. So the pentose phosphate pathway here. Oh my God, this is really small on my screen. I really should have cut that down. So this right here is your phase 1. And then we go into phase 2 right here. Good God, can you guys even see that? No? Okay. Well, I'll have a little thing where we discuss it more in the breakdowns. But yeah, I really need to work on that figures. Let's see. Jesus. Can you see it now at all? Yeah, okay. I might have to cut that figure up and yeah. Anyways. You get the overall, we've started talking about this, and I'll break it up into the text parts and we'll be able to talk about it that way. Yeah, damn. Okay, so the glucose 6-phosphate dehydrogenase, this is going to initiate the oxidative phase of your pentose phosphate pathway with the conversion of the glucose 6-phosphate into the 6-phosphoglucano delta lactone. This will yield the NADPH and the H+, specific for NADP+, highly specific for that. And so this is actually kind of key to this. So you need the NADP+. It's not going to take things like NAD+, okay? And specifically, this is one of the ways you're going to generate the NADPH, which you need for things like oxidative stress and the other pathways we talked about. So lactonase, this is going to hydrolyze your 6-phosphoglucano delta lactone to the 6-phosphogluconate. The 6-phosphogluconate dehydrogenase is going to oxidatively decarboxylate the 6-phosphogluconate to the ribulose phosphate. This will yield the CO2 and the NADPH. NADP plus is your electron acceptor. This is one of those electron acceptors we had listed out and talked about as being an electron transporter. It's funny, I never think about my computer at home actually having pretty good screen size and stuff, but it's times like this that I encounter this that I really go, oh shit. Screen here is much smaller. Okay. The oxidative phase of the pentose phosphate pathway will yield your 2 molecules of your NADPH, and this is actually that part of that pathway that we would have talked about, okay? One of the things I do kind of want to point out, and I don't know if I've really pointed this out yet, is that while these do look like linear pathways when we're looking at them here, it's because we've simplified them so much. We've removed other side paths. If we incorporated that web back in, it would come back around. Okay? It's very rare to find something that is not recycled at all eventually. Okay? But some of these are very energetically unfavorable, and so you don't have direct recycling. Because direct recycling would just take far too much energy. Okay, so this is the glucose 6-phosphate. It's going to react via the enzyme glucose 6-phosphate dehydrogenase. This tells you a lot about that because we're going to dehydrogenate. And so we remove the oxygens across this, what will end up being a carbonyl bond, this oxygen-carbon bond here. And this is going to give the reduction to the NADP plus to NADPH. So we're oxidizing this, we're reducing this species. That's the 6-phosphoglucano delta lactone. Lactone being this group right here. Okay, that's the ON ketone, right? ON. And the delta being the position. You know, the alpha, beta, gamma, delta. Yeah. So this is then going to react with the enzyme via the enzyme lactonase, the lactonase, the enzyme that works on it. This is going to use a water going to H+. This is actually going to split this up, and we're going to get the carboxylate group right there. That being the carboxylate group. Notice again, this right here is this group right here. This carbon is this carbon right here. And this right here is the carboxylate right there. Okay? This is that, remember where we did those reactions breaking apart the esters? Carboxylic acids? Same thing. Just we don't get a carboxylic acid out of this because we're in a buffer solution. We get a carboxylate. So this gives you the 6-phosphogluconate, 8 carboxylate. The NADP plus is then going to go to NADPH because we're going to go ahead and oxidize this right here, giving this the reduced form. This enzyme is going to be 6-phosphogluconate dehydrogenase. Notice the extreme similarity here because, again, it does very similar stuff, right? So this is going to give us the ribulose 5-phosphate. By the way, that being the 5-membered sugar that's going to have phosphate on it, that this pathway will end up being named. And a CO2 here, which is the fully oxidized carbon, by the way. And so, surprisingly efficient. The phosphopentose isomerase is going to isomerize the ribulose 5-phosphate into the ribose 5-phosphate. And so right here we have the ribulose 5-phosphate, this one being the ribose 5-phosphate. Notice that we have transferred, instead of having a ketone, we're going to end up with an aldehyde. Okay? And so we went from a ketose to an aldose. That's the isomer. Okay. Same groups overall, just different isomer. The transketolase and transaldolase are going to catalyze 3 successive reactions. And so this right here is the transketolase. We had kind of mentioned this stuff before and looked at it. So this is the C5 plus a C5. It's a transketolase. This gives you, again, C3 and C7. Notice, again, they always have to have the same number overall. The transketolase here is going to react C3 with C7. Notice that's the product actually here. This will give us C6 and C4. This is another transketolase. We got the C4 from right here and the C5 from up here. And so that's going to then give us the C6 and C3. And so notice we actually have things plugging back into these equilibriums from each other. Okay? For example, C3, C3, feedback. Okay? All right. So the net result of these reactions is actually a conversion of 3 pentoses into 2 hexoses and 1 triose. And this is what's usually considered the overall net result of this. But again, it does not have to be, if you are removing one of these things, it's going to keep populating that thing, right? Because they're stacked equilibriums, okay? But this would be if you took these equilibriums and just added them all together. Notice you'd have the C3 canceling each other right there. Okay. And so this would give you the overall reaction. So the trans-ketolase, and this is pretty common because remember, the most used sugars in our body are actually the C6s, right? That's the glucose that we're burning through all the time, okay? And so it's very common for that to end up going that direction. The trans-ketolase is going to catalyze the formation of glyceraldehyde into the 3-phosphocetoheptylase, 7-phosphate from 2 pentoses. First of the 3 linked reactions. Now I would like to point out that there's something a little bit special about the C3s here. They're actually pretty easy for you to generate and they're very ubiquitous. Okay, so your C3 sugars are actually end up getting fed into a lot of stuff to make other things. So whenever you see a C3 sugar and a C6 sugar, those are kind of your ubiquitous things. Your C5s actually make quite a few things, but they're less ubiquitous compounds. The trans-ketolase is going to catalyze formation of glyceraldehyde 3-phosphate. Xylulose 5-phosphate with ribose 5-phosphate. Here we have that trans-ketolase. This is going to give you your glyceraldehyde 3-phosphate. You may recognize that from many other pathways. And then this is the cetoheptylase 7-phosphate that we had mentioned before. So the trans-ketolase substrate is going to be a ketose with the hydroxyl group at the C3, having configuration of xylulose rather than ribulose. So the phosphopentose epimerase, this is going to catalyze the conversion of the xylulose 5-phosphate into the epimer ribulose 5-phosphate. And so this is your ribulose 5-phosphate. This is the phosphopentose epimerase right here. And what you notice is we're just flipping this group right here. Okay, that's the only difference. And that's going to give you your xylulose 5-phosphate. Okay. And everybody remembers these structures, right? Okay. Yeah, these are coming back, coming out towards you. Okay, that's that Fisher projections. Okay, so the trans-allose is going to catalyze the formation of your 6-carbon and 4-carbon sugar from a 3-carbon and a 7-carbon sugar. This is the second of your 3 linked reactions that we had talked about just a bit ago. We had been a lot less clear on what exactly happens in these, but now we're actually clarifying specifics on some of these. Again, these are one version of it. The glyceraldehyde 3-phosphate is going to react with the cetoheptylase 7-phosphate. This is going to give you your fructose 6-phosphate and etherose 4-phosphate right there. By the way, if you want to know exactly which groups go where, this is the color coordination. See that? So like this came off of this one. That came off of that one. We're not going to go super specific in the mechanisms. It's just, this is not organic chemistry. That would be extreme, and I wouldn't have time to even begin to do that. Yeah, same thing here. See the purple group right there, comes there. Red. Yeah, and the blue. Okay, the reason this was purple, by the way, blue, red, on, is these are actually self-consistent within this stuff. Okay? So the purple came from the purple. Yeah. So the trans-ketolase catalyzes the formation of the 6-carbon and 3-carbon sugars from the 4-carbon and 5-carbon sugars. This is the third of that 3 linked reactions we had talked about before, where we had an overall reaction for the whole thing. And this is the etherose 4-phosphate in this case, xylulose 5-phosphate, the trans-ketolase gives you fructose 6-phosphate and glyceraldehyde 3-phosphate, and that's that 3-membered sugar, by the way. So your sum of your reactions that are catalyzed by these trans-ketolase, phosphopentose epimerase, and trans-allylase is this right here. We have 2 xylulose 5-phosphates plus a ribose 5-phosphate, giving you 2 fructose 6-phosphate and a glyceraldehyde 3-phosphate. Remember, this is the overall results you do need multiples of some of these to make it go. This is that overall thing. Okay. Overall reaction. So the xylulose 5-phosphate can actually be formed from ribose 5-phosphate via the phosphopentose isomerase, the phosphopentose isomerase. And so the net reaction, if we start from the ribose 5-phosphate, which is what we considered the starting point at the beginning of this, would be that final overall reaction we'd seen on that previous slide, which had the 3 riboses. Okay? So 3 ribose 5-phosphate going to 2 fructose 6-phosphate and a glyceraldehyde 3-phosphate. Remember, that is the overall reaction, though. That does not mean that this is the only sugars that are involved in that entire pathway. So this is the pentose phosphate pathway given in a nice tabular summarized format right here. And so this is the oxidative phases right here. These are the enzymes that do those reactions. These are the non-oxidative phases right here, and they're the enzymes that do those reactions. Note, by the way, you can actually tell which one's which very easily because these have dehydrogenase, a lactonase, and dehydrogenase. And then these ones right here are rearrangements and chopping parts of one on putting on the other, right? And so you have no overall oxidative activity. You're just rearranging the sugars. Okay, and so this is the trans-ketolase and trans-allylase are going to stabilize your carbanic intermediates via different mechanisms. Your trans-ketolase is going to transfer a 2-carbon unit that contains tightly bound thiamine phosphate. This is your TPP. That's the prosthetic group. The trans-allylase is going to transfer a 3-carbon unit and does not contain a prosthetic group. Each of these carbon units is transiently attached to the enzyme in the course of the reaction. Characteristic of double displacement reactions, actually. So this is one of those nice places where we give you specific examples, right? But it's actually a wider spread motif that you can apply to many reactions, okay? So this is the trans-ketolase reaction. It's going to take place on the thiamine phosphate. Here's our TPP. So the first step of this is it's actually going to lose H plus here. That loss of this H plus right here is going to leave behind a carbanion right there. Okay? And so that's that very reactive intermediate species. This is what's going to act. Anybody recognize that reaction, by the way? Remember our carbanion, which is a strong nucleophile attacking our electrophilic carbonyl? Yeah, it's just that. And the only difference here is TPP is a prosthetic group, so it doesn't go off and drift off and, attack shit willy-nilly. It can only attack the stuff that can fit in, right? Yeah. So this is going to attack your carbonyl here. This is the ketose substrate. That will allow the electrons to go up onto this oxygen, which is always that first step whenever you have those carbonyls, right? And in this case, what we'll have is an H plus is going to go ahead and react with that, what used to be the carbonyl is now the oxide. This is actually the addition compound, by the way. And the next part of this mechanism is at this point we have 3 alcohols here, all next to each other, trial. And that's going to be pretty reactive if we have this positive charge here, right? And so what's going to end up happening is this proton here is going to be a bit more acidic than it normally would be. It's going to lose its electrons into this bond, making a carbonyl. This is then going to have the electrons transfer from that carbon here over into this bond between the carbon and this carbon over here on the TPP. And this double bond is going to move its electrons up onto that amine. Now note that the aldose substrate is going to be what's going to end up getting reacted here in this next step. And this is going to go into the aldose product plus the H plus here. And so this is then going to give you your activated glycoaldehyde. And so note that we've split this up here. And we now have this double bond. See, this has gone away. We now have this double bond here, right there. Okay. So the aldose substrate is going to be the one that the carbonyl is going to react with that pair of electrons right here. Again, this is more reactive than it would normally be because it's stabilized by this group, which has a lot of resonance properties, right? Has many forms it can take. So that's that activated glycoaldehyde. These are going to react then, moving the electrons up onto that oxygen. This is going to move that lone pair into a bond. This double bond is going to be the one that acts as your nucleophile, right? This is going to give us this activated, this form right here. Note, by the way, we do have an H plus that needs to come on here to complete this reaction. That's up here. Remember, we had the alkoxide up here. Yeah. Okay, and so the next step is actually, again, we have these two alcohols. If this one loses its proton, we go back to this. If this one loses its proton, we can go forward. In which case, we lose the proton. The electrons that were in that bond are going to join and form the double bond here. And then this is going to give the electrons back to reform the TPP. This right here is going to take on an H plus when we go forward. And that's because we have made that, it would have been a TPP carbanion, but we've actually shortcutted to TPP, going back to this TPP right here. Note, by the way, though, that if this H plus had not come on, this would have been the TPP carbanion, which would have reacted with this very easily. Okay? Okay, that's the ketose product, excuse me. Notice that we went from this ketose substrate to this ketose product where we have exchanged these carbon groups. We've translocated them. Ooh, ah. So impressive. It is. So the step one for the trans-ketolase reaction is going to be the C2 carbon of the bound TPP ionizes to give the carbanion. That's what we had seen before. The carbanion of the TPP attacks the carbonyl group of the ketose substrate. Step 3 is the cleavage of the carbon-carbon bond, frees the aldolase product, yielding an activated glyceraldehyde, join TPP. Notice that's one of the products we had talked about before. It made 2 products. That's one. Step 4, the carbonyl group of the aldolase aldose acceptor condenses the activated glyceraldehyde. This is going to form a new ketose. Step 5, the ketose is going to be released from the enzyme. This will free the TPP from the next reaction cycle. This is the trans-aldolase reaction. This is going to involve a shift base intermediate here. And so what's going to happen here is we'll have the lysine on the enzyme here. It's going to then react. Remember, this one does not have a prosthetic group, okay? So the ketose substrate is going to react with this. Notice that this is what type of reaction? If we lose water, dehydration, right? If we go reverse, it's hydrolysis, yeah. Yeah, the reverse would be the hydrolysis. Yeah. And that would be splitting this up with water, right? So this actually is a shift base. You may remember that from your organic chemistry, actually. So shift bases, that's that nitrogen double bonded to stuff right there. That's the shift base, okay? I know, that damn organic chemistry shit, it's never going away, is it? Okay, so the next thing that's going to happen here is we're going to protonate that shift base. We're setting it up for the next reaction, making it more reactive intermediate. And so this is the protonated shift base. What happens next is that proton on there is pulling electron density. And so it pulls the electron density from the double bond. That gives this carbon a positive charge, which then pulls the electron density in the bond next to it. and making a double bond there. This actually pulls the electron density, making this carbon positive, and so it pulls the electron density from this proton here, releases the proton, giving us a carbonyl double bond. So release the proton, the acidic proton, gives you the aldose product here. That's one of the products that can be then released and drift away. And it gives you this reactive, what will be a intermediate here. Now, the next thing that's going to happen is we're going to take in an aldose substrate. This is going to have the carbonyl here. Remember, the carbonyls can react with remember, this can act as electron withdrawing group here, or electron donating group, right? Because it has that lone pair up here that can bleed electron density into here. And so what you're getting here is that more reactive double bond. Remember, double bonds can actually act as nucleophiles. And so we're going to react as a nucleophile, attacking this carbonyl. This is going to put the electrons up on this oxygen here, like those reactions always end up doing. And it'll join a bond to these carbon here. Note that we're going to pull the electrons into a double bond now. That's going to make this a positively charged shift base, that protonated shift base again. See that? We're regenerating that. We just have this aldose substrate attached now. So the next part here, this does need to take a proton on to this alkoxide that would be generated for it to be stable enough to go forward on the other side. Okay? And that's where that proton goes. So the next step here is going to be losing that proton on that protonated shift base, regenerating the shift base. Okay? And so we regenerate that shift base, the double bond there. Next is going to happen. This is the what? Hydrolysis. You got it. And so we're going to hydrolyze it. Okay? And so we hydrolyze it, cut that off. Notice the carbonyl here from the hydrolysis. And we regenerated the lysine. Okay? Note this does not actually curve back around into a cycle because we do not end up with the same thing here as there, do we? No. So, the trans-aldolase reaction, the step one is the shift base forms between lyse residue and the trans-aldolase and the ketose substrate. Step two, the protonation of the shift base occurs and the bond between C3 and C4 is split. Step three, deprotonation leads to the release of the aldose product, leaving a 3-carbon fragment attached to the lyse. Step four, a suitable aldose binds. Step five, protonation allows the formation of the new carbon-carbon bond. Step six, deprotonation occurs. Step seven, hydrolysis of the shift base releases the ketose. This is the overall portions that we were talking about with those previous reactions. So for the trans-ketolase and the trans-aldolase, a carbanion intermediate is actually stabilized by these resonance forms here. And so this is your TPP prosthetic group. Notice it has the double bond form here, but it actually has these negative and positive charges like this as well. And so that's that resonance form. Remember the double arrow like that, where it had two sides? Meaning resonance, meaning the overall structure is a combination of those, right? Yeah. And these are why those reactions occur like they do. So that's that negative charge that's attacking those carbonyls, right? That's your nucleophile right there. That's why it's a nucleophile, okay? And it's that resonance form that gives that positive charge. This is that protonated shift base right here. And note, when it has that proton on there, this actually gives this two different forms here. The form where it's the double bond carbon, like we had looked at, but it's not actually all just neutral there. This right here has a partial positive charge, which is that, and this one has a partial negative. And again, that's that negative charge on there that's going to attack the carbonyl. That's why it directs to this carbon, not this one. Okay? That's why we get that final product we do. Okay? So the glucose 6-phosphate is going to be metabolized by both the glycolytic pathway and the pentose phosphate pathway. The fate of the glucose 6-phosphate is controlled by the cytoplasmic concentration of the NADP+. The first reaction, the pentose phosphate pathway, is the dehydrogenation of the glucose 6-phosphate by glucose 6-phosphate dehydrogenase. This is the rate-limiting step of the pathway. This takes the majority of time of the pathway. The reaction is essentially irreversible. It cannot go backwards, typically. Low levels of NADP+ are going to limit the dehydrogenation of the glucose 6-phosphate because it's needed as the electron acceptor. So this ensures the NADPH is not generated unless it's needed for reductive biosynthesis or protection against oxidative stress. Or another way of saying that is we have an overall equilibrium here. It's the rate limiting step. We need the NADP+ to do this. So if we have not used the NADPH up enough to have NADP+, so this reaction goes at a decent rate, it's slow. The rest of the stuff's not going to go. Okay. All right. So the pentose phosphate pathway. This can be operated in 4 distinct modes. Much more ribose 5-phosphate than NADPH is required. The needs for the NADPH and ribose 5-phosphate are balanced. If we need much more NADPH, then the ribose 5-phosphate is required. Both NADPH and ATP are required. And so these are the 4 different switches, as it were. Okay? Think of it a bit like you're studying. You may be studying 4 classes, right? And you need to balance the needs of those 4 classes' studying habits, right? And so some things can overlap a little bit, and you get a little bit of studying for, say, cell biology and genetics when you're studying biochemistry, don't you? Okay? And so there's a little bit of overlap between those. The same thing's going to happen here. Okay? These pathways will give you slightly different modulations. I think I need to expand that figure out. Okay, can you guys see it okay? Okay. So these are the 4 modes, okay? And honestly speaking, these 4 modes, if you just understand the pathway, you can pretty much tell which one you need and don't need by what the pathway makes, right? Okay, so mode 1 is going to have glucose 6-phosphate go to fructose 6-phosphate. This is then going to give you fructose 1,6-biphosphate and ribose 5-phosphate, though it needs one of these end products to do that, okay? And so next, that fructose 6-phosphate goes to that fructose 1,6-biphosphate, as we said before. That gives you, split up like this, the dihydroxyacetone phosphate, glyceraldehyde 3-phosphate. You guys all recognize that, right? Glycolysis. The dihydroxyacetone phosphate, just like glycolysis, can go into the gap, the glyceraldehyde 3-phosphate. This glyceraldehyde 3-phosphate is actually what's necessary in this step right here. We went from fructose 6-phosphate to the ribose 5-phosphate. And this is one of the primary products of this. And typically this is when you need, like it says here, we need a lot more 5-phosphate than we need the NADPH. Okay? So we have very little NADP+, we have plenty of NADPH, and so we need ribose 5-phosphate is getting consumed. We need to replenish that. Okay? Mode 2 is going to, if you look at this, we're generating a lot of NADPH, and we're also generating ribose 5-phosphate, aren't we? Okay? And so we have a lot of NADP+. This is going to be glucose 6-phosphate, consumes 2 NADP pluses, and releases the CO2. This gives you 2 NADPHs, which can now be used to do reductions other places and carry electrons there. This will give you ribose 5-phosphate. This will then be converted to ribose 5-phosphate, which again is one of those, we need ribose 5-phosphate. Okay? Notice, by the way, you produce one of these as well. But what else do you notice about this? What else do you consume in this pathway? You need NADP pluses, don't you? Yeah. Do you need any of that there? No. So you don't have the NADP pluses because you have plenty of NADPH. Mode 1. If you need both, mode 2 will work. Okay. Mode 3 is going to again take, we're going to start right here actually. So we have the glucose 6-phosphate. This is going to go to, notice by the way, this one is a cycle, self-contained cycle. Though you do need to continuously input NADP plus. Mode 2, by the way, is right here. Can everybody see that? This is mode 2. Okay. What do you notice about this part right here, by the way? It's the same stuff as mode 1, right? Yep, we're going in reverse. Okay, so that's one of the easier ways to sort of remember this, okay? And so what you're going to be getting here is the glucose 6-phosphate, again, with 2 NAD pluses, gives you 2 NADPHs. This is going to lose the CO2, give you ribose 5-phosphate. We then are going to make ribose 5-phosphate, just as you would in mode 2, okay? But we don't need the ribose 5-phosphate. Nothing's consuming it. It's just sitting there. And so it starts feeding into these reactions here. And so we get a back feed of basically what would have been mode 1, okay? And so our ribose 5-phosphate back feeds to make fructose 6-phosphate. It back feeds to make glyceraldehyde 3-phosphate. The glyceraldehyde 3-phosphate builds up and it can go to dihydroxyacetone when we have one of each of those. Those are going to react to form the fructose 1,6-biphosphate. That's going to then give you that combined together with this right here to give you the fructose 6-phosphate. This will then give you the glucose 6-phosphate. Notice we have recycled these carbons that were remaining, so that we can keep pumping out more and more NADPH. Okay? And that's where you just need that. You have plenty of the other. Notice again, though, that you don't want to continuously just produce something over and over and over and over and never recycle it, right? If this is not getting used up, it's going to backfeed into these so you can keep producing NADPH, which presumably you're using up and getting NADP plus to keep going in this pathway, okay? This is mode 4 right here. This one's kind of cool because it joins this pathway with other pathways that you have seen before, okay? So we're actually starting to link this pathway up into even more stuff. Remember, mode 4 is both NADPH and ATP are required. You don't need necessarily the you don't need necessarily like a ribose 5-phosphate. It does produce the NADPH like before, but we're going to be a bit hampered by it because we're not going to recycle as much, okay? But on the other hand, we're going to produce ATP, and we're going to actually produce quite a bit of it because we're actually ending this pathway prematurely. So mode 4 is really just mode 3, isn't it? See that? Yeah. But the only difference here is that we have linked this up, just like we did with glycolysis, into our energy production pathways that will eventually lead into the citric acid cycle and oxidative phosphorylation. Okay? And so we're going to pyruvate right here, aren't we? See that? Pyruvate going to go to acetyl-CoA, going to go to citric acid cycle, going to make lots of ATP, right? Yeah. And so we don't backfeed in this all the way in back to this glucose 6-phosphate. Okay? So glucose 6-phosphate, again, mode 2. See that? And then we get into the ribose 5-phosphate. This does backfeed into the same stuff as here. But in this case, that glyceraldehyde 3-phosphate's getting used. And so it can't join back together and go up. Okay? What instead happens is all those equilibriums are going to tilt towards that and start draining out. The ribose 5-phosphate goes to glyceraldehyde 3-phosphate, consumed for ATP and pyruvate. Pyruvate going off, getting used up, right? Citric acid cycle eventually. The fructose 6-phosphate is again going to not have this backfeed going up. It's going to keep getting drained out, right? Into this direction on that. So this is going to generate the fructose 1,6-biphosphate. It'll break apart. The glyceraldehyde 3-phosphate gets consumed. So this dihydroxyphosphate ends up going over to that in that equilibrium. Okay? And that's because you're generating lots of ADP and AMP. Those are then consumed to make the ATP, right? And you're using lots and lots of energy. Okay? Okay. So mode 1, you need much more ribose 5-phosphate than NADPH is required. Occurs, for example, when rapidly dividing cells. You need the ribose 5-phosphate for the synthesis of your nucleotide precursors of DNA. Glucose 6-phosphate is going to be converted into the fructose 6-phosphate F6P. Glyceraldehyde 3-phosphate, GAP, normally referred to as GAP, is going into the glycolytic pathway. The trans-allylase and trans-ketolase will convert 2 molecules of the F6P and 1 molecule of GAP into 3 molecules of the ribose 5-phosphate. Your stoichiometry of mode 1 is this right here. And so you don't need a lot of electrons when you're making lots of DNA, right? That's not the main thing you need. Okay? You need the damn sugars, right? And so we're making that sugar. The glucose 6-phosphate, this is going to be, you need ribose 5-phosphate and NADPH. We're in balance in this case, and so we want to make both. And so the glucose 6-phosphate is processed to 1 molecule of the ribose 5-phosphate, generates that 2 molecules of the NADPH. Ribose 5-phosphate is then converted into ribose 5-phosphate. Your stoichiometry of mode 2 is this right here. So we need a lot more NADPH than the ribose phosphate, and that's mode 3. One of the major times this would occur is when the adipose tissue requires a high level of NADPH because we need to synthesize lots of fatty acids. And so we're building up fat stores. Okay? The glucose 6-phosphate is going to completely oxidize to the CO2 through 3 groups of reactions. We have the oxidative phase of the pentose phosphate pathway. This is going to form 2 molecules of the NADPH, 1 molecule of the ribose 5-phosphate. The ribose 5-phosphate is converted into the F6P and GAP by the trans-ketolase and trans-allylase. The glucose 6-phosphate is resynthesized from the fructose. Remember, this one recycles. Okay. This is the stoichiometry of mode 3. I'm just going to let this be a summary of that. Does anybody have any questions of that? Okay. Remember, this one recycles, so it can really pump the NADPH out. Okay. Remember, stoichiometry, you go minimum of 1. You don't have to do halves normally, right? So, for example, one of these glucose 6-phosphates ends up pumping out 12 of these NADPHs because you keep recycling parts of it. So it actually deals out a bunch. Okay, and that's why mode 3 is extremely good for that. The tissues with active pentose phosphate pathways are these tissues right here. We have the adrenal gland for steroid synthesis, liver for fatty acid and cholesterol synthesis, testes for steroid synthesis, adipose tissue for fatty acid synthesis, ovary for steroid synthesis, testes and ovary being the 2 genders, of course. typically. Mammary gland for fatty acid synthesis, and red blood cells for maintenance of reduced glutathione. Man, I am totally, if I monetize my videos, I'd be screwed this video, wouldn't I? Like, I covered global warming and now that bullshit. There's bullshit things in there. Not global warming in general being bullshit. I covered there's two genders. Oh my God. I'm in trouble. Mode 4, both ribose 5-phosphate and NADPH. These are required in mode 4. Ribose 5-phosphate is formed from the glucose 6-phosphate. This can be converted into pyruvate. F6P and GAP are going to be derived from the ribose 5-phosphate. These are going to enter the glycolytic pathway rather than reverting to the glucose 6-phosphate. The pyruvate that is going to be formed by oxidized to generate more ATP, or it can be used to as a building block in a variety of biosynthesis. I know I did kind of stress the ATP production of this, but remember, pyruvate's a ubiquitous molecule that shows up just all over the place, right? So this could end up being a building block for a variety of different substances. You could end up doing this because you're moving things around and healing. You could end up doing this for a variety of reasons. Okay? Pyruvate does not actually have to go into the citric acid cycle, and even if it does go in the citric acid cycle, remember, many of those products in the citric acid cycle can go off into other pathways. Okay? So this is a very ubiquitously useful molecule, very ubiquitously useful pathway. The stoichiometry of the mode 4 is this right here. Again, remember that this does have some degree of ability to have some recycling going on, okay? And various different pathways in here. Remember, this is coming back here. Okay, the pentose phosphate pathway is required for rapid cell growth. Rapidly dividing cells, like cancer cells, for example, require the ribose 5-phosphate for the nucleic acid synthesis and NADPH for the fatty acid and membrane lipid synthesis. Rapidly dividing cells are going to switch to aerobic glycolysis to meet the ATP needs. They're going to divert the glucose 6-phosphate and glycolytic intermediates through the non-oxidative phase of the pentose phosphate pathway to generate ADPH and ribose 5-phosphate. The pyruvate kinase isozyme, PKM, this is going to facilitate the diversion of glycolytic intermediates into the non-oxidative phase. PKM has a low catalytic activity, meaning it doesn't have a huge speed up. Glycolytic intermediates accumulate and enter the pentose phosphate pathway. Diversion of the phosphorylated intermediates is going to further enabled by the inhibition of triose phosphate isomerase via the phosphoenolpyruvate, which is a substrate of PKM. The Kelvin-Benson cycle is going to begin the fixation of CO2 and proceeds to the NADPH in the synthesis of glucose. The pentose phosphate pathway is going to begin with the oxidation of your glucose-derived carbon atom to CO2, generating NADPH. In the photosynthetic organisms, many of the enzymes are going to be common in the two pathways, and this is why normally we keep these two things kind of separated, right? And we try to do that because many people are going to work on non-photosynthetic organisms, don't give a shit about them, and many of the ones that do work on photosynthetic organisms can kind of ignore the other ones as a, biochemistry. And so it just tends to be less important between the two. This is where these two things are just joined at the hip, right? The reduced glutathione, GSH, this is a tripeptide with a free sulfhydryl group that's going to combat the oxidative stress by reducing the ROS to harmless forms. Remember, your oxidative stress is going to be a generalized concept where we're going to take these oxidized species that would oxidize other species, and we will reduce that overall oxidative state stress in the entire system, right? Okay. So the oxidized glutathione, this is GSSG, must be reduced to regenerate the GSH, which is back there. That's the reduced form. So remember, oxidized, GSSG, reduced, GSH. Okay? H being reduced, right? Losing H's means oxidized. Okay, so the reducing power is actually going to be supplied by the NADPH that's generated by the glucose 6-phosphate dehydrogenase in that pentose phosphate pathway. That's why we kept referring to that. This, by the way, is what we're talking about here. And so this is the glutathione. This is the reduced form. You can very easily tell that's the reduced form because of that right there. See the SH? There we go. And when it's the oxidized form, what would you expect? Another one of these, right? So you'd get another one, and you'd have a disulfide bridge. Okay? And that's the oxidized form. All right, so the pemiquin, this is the first synthetic anti-malarial drug. This is introduced in 1926. Some patients develop drug-induced hypolytic anemia due to a deficiency of the glucose 6-phosphate dehydrogenase. This deficiency results in the scarcity of NADPH in all cells, most acute in the red blood cells because they lack mitochondria. By the way, if you want to see just the utterly ridiculousness of the world and how things work, watch some videos on malaria, okay? And what's been done and how many people die of it a year and all that, okay? The drugs for this can be produced for pennies and are produced in sufficient quantities to treat the entire world, and nobody ever has to die from malaria. And we could stop it. We could stop 1 million deaths by malaria a year with a couple $1 million. Literally dollars per person. And it's actually kind of impossible because every time it gets tried, the shipments don't make it to where they're supposed to go. It's nothing to do with money. It's so fucked up. I'm not joking. This is one of the most fucked up situations on the planet. Okay. Anyways. So the most acute in red blood cells because they lack mitochondria, and that's the pemiquin right there. So primaquine. This is primaquine is an anti-malarial that's closely related to pemiquin. This is widely used in malaria-prone regions of the world. The drug will induce the hemolytic anemia, and it's seen in patients deficient in the glucose 6-phosphate dehydrogenase, and that's, again, primaquine. The fava beans produce a pyrimidine glycoside, and so vicine is a pyrimidine glycoside of the fava beans. These are the fava beans right there. People that are deficient in the glucose 6-phosphate dehydrogenase actually are going to suffer the hemolysis, from eating these fava beans, just like they would from these drugs here. And what do you notice about this? Vicine right there. It looks a little bit like that, doesn't it? So the peroxides, these are going to be reactive oxygen species that can damage membranes and other biomolecules. These different ingredients, the pemiquine, primaquine, and vicine, all generate peroxides. And so, again, these are all oxidative agents. These will generate peroxides. That's that oxidative load we were talking about, right? And this is why you need to fight the oxidative load. And one of the main ways you do that is you generate NADPH, right? Which is then used to lower that oxidative load. The glutathione peroxidase is going to eliminate the peroxides. It's going to use glutathione as a reducing agent, and that's that glutathione that we talked about just a bit ago, that GSH. And so 2 GSHs react with a peroxide with this enzyme doing so, and this gives you GSSG, an H2O, and an alcohol. In red blood cells, the glutathione reductase is going to use the NADPH to regenerate the reduced form of your GSH. And so right here, again, we have the oxidized GSSH, has two of them right there at the bridge. And then we have NADPH. This is going to put the electrons on there to regenerate those two GSHs right there. So I'm sorry, but give me a moment.